So
I was here at 8:30 in the morning.
So 2:30, the class data is 2:30. Joshua will be presenting a practical on assignments for the last 20, 30 minutes. So since it's delayed, he may have to keep going. So, I have to take a Zoom call at 2:30. So I will exit at 2:30, take a call. Not that it matters to you guys.
People can vote with their feet.
Okay. So I'll wait till more religion for this stage. So there's pizza, like for class?
No, not those.
Why does that class have pizza? And not ours?
They're nicer.
Every class they have pizza together. Can't be it. It was a seminar.
If we get feedback form for this class, I might like that. Yeah.
You guys will fall asleep today. Can't do that. So we're just waiting for my students who are off residence. Well, theoretically, they should know all of this, so. Yeah, sorry, you asked, tomorrow. I'll email you. Tell me which times work best for you. Okay, so I'll email you. Why not? Because I'm wrong. You're happy about that? Why? You don't like those meetings? Oh, no, yeah, you get postponed, that's all. There's no other day to do it until we get back.
When you fly out.
Tuesday. And back Friday. Thursday. Tuesday, unfortunately, because of all these other things. I wish I could say next year, once I'm done with this job, Art, I think 3 years is enough. I mean, at least it's up and running, so. As I told you, it'd be great to meet. Probably we're going to see to discuss teaching.
The feeling as to what paper from last June to this June. I should highlight the progress of the CEO. And it was picked on.
There's few. There's the Carboneo relaxation R1 road that Sophia and Shabani had in JMR, and it was the cover. And that was like the measuring tautomeric states and using the carbonyl on nitric acids when previously it was done on proteins by all your former students, basically. And then there's a paper that just came out in Cell, where we did a bunch of proton cell experiments on many different variants of TAR and showed that the exchange kinetics dictates the cellular activity. So if you want more if you want more like technical, the carbonyl is more technical and the cell is maybe more biological.
We'll look and see when last year we've already talked about.
I don't think so. That was like Supriya just did it in a short time frame. But maybe we had proposed it at some point. So Rachel, any update on their? Yeah, I think I should start reading like. What did it say? No write for them today. Right back. Oh, there they are.
Sorry.
No. Okay. Good. So, Justin, you're ready with your slides, right? Towards the, so that class ends at 1:30. Sorry, 2:30. So let's say that at 2:10, you can jump on and hopefully you know how to do the.
Yeah.
Okay. Ryan is a new student rotating in my lab. So he's delving into some NMR. Ryan, that's Professor Al Palmer that I mentioned, whose book you're supposed to study from A to Z. If you have any questions about the book, you can ask it. Okay, so I'm going to continue on with the application of NMR for the study of nucleic acids, and we've been focusing on RBC measurements and their utility in defining global aspects of nucleic acids, and we talked about this idea that you can take fragments of an RNA. So the nice thing about nucleic acids is that they're built up of, you can think of them as Lego pieces. They have robust building blocks. In the case of RNA, the A-form helix. And if it doesn't have mismatches to a good approximation, you can model those helices assuming an A-form geometry. For the DNA, you can assume a B-form geometry. These are approximate geometries. And then you can solve for the order matrix equation that we discussed. Oh, Rachel, let's see these names. So we can, if I have more than 5 independent RNC measurements, and I know the structure of the A-form helix, I can solve for the 5 elements of the alignment tensor, which tell me about how the object is aligned relative to the magnetic field. We discussed that we have the order angle defining the orientation of the entire object relative to the field, and the 2 order parameters describing the extent of alignment and the asymmetry of alignment. And I can determine the average orientation of 2 helices by insisting that their order tensor frames ought to be superimposed in a common direction, just like I can determine the orientation of countries on a map. If I took Italy and told you where the North Pole is, and I took the US and told you where the North Pole is, you can determine their relative orientation by insisting that the pole directions that they both point north in common. Here we are doing this 3 dimensions because we have north, south, east. There's a 3-dimensional frame with z, x, and y directions. And so what we can do is basically rotate each domain into what we call a principal axis system, where s is along z, s or y along y, s is along x. That's how this fragment aligns relative to the magnetic field. The field is going up and down. This is how this fragment on average aligns relative to the field. And by stitching these two together, I can say that's my average orientation of the 2 parallel domains. And now there's one interesting problem here, which is that there's no distinction, unlike north and south. I can tell the difference between north and south. Here, actually, whether z is pointing up or down, or whether x is pointing, y is pointing this way or that way, or x is pointing in or out of the board, is something that is degenerate. It's not something that is uniquely defined. So when I assemble those 2 fragments, I can fix one of them, and this other one can have 4 allowed radiations. This one that I'm showing here, but also 3 additional ones involving 180 rotations around x, y, and z. So I can't tell the difference whether SDC is pointing up or down. There's no directionality to it. And so I have a degeneracy, a 4-fold degeneracy arising from the fact that I cannot determine whether something is aligned up or down. So there's an ambiguity. Any thoughts on how you might resolve that degeneracy? Now remember, I put the sample in an ordering medium, let's say phage. Phage dictated the alignment of the molecule, and then I got myself an alignment tensor. Imagine you had control over this experiment completely. What would be a way to overcome this degeneracy? What would you love to be able to do? Any thoughts? Okay. Great. Change the alignment tensor. Great. That's right. Did you read the paper already? That's good. Okay, so if I take my virtual parallel objects, domain 1 and domain 2, I align the two, I superimpose the tensors. Here's one solution, but I can take the parallel object and flip it 180 degrees about 3 different directions, either X, Y, or Z. So these are the 4 allowed solutions. These are the 2 domains of a protein or RNA. And so if I now generate a new alignment tensor, which means I change the orientation of the alignment somehow, I will get another set of 4 solutions. But if the new alignment tensor is not coincident with the original alignment tensor in all 3 directions, 3 of these solutions, one solution will be in common, the correct solution, but the other 3 will not overlap by definition, because by changing the alignment tensor orientation, the rotation axis about which I'm generating the solutions has been altered. These C don't match C, whereas this one matches. So by changing the alignment tensor, clearly I'm getting more information. And the key point I want to make here is that in NMR, you often can get more information by measuring more things in the Hamiltonian. But sometimes you can also alter the sample to get more information. In fact, a lot of times you can not only simplify the experiments, but also getting more information. And here's an example. If I just had an actual way of changing the overall alignment tensor by, for example, changing the ordering medium, I could get another alignment tensor, another set of RDCs. And now when I solve for the degeneracy problem, I find one common solution. Now, the problem with this approach is that while changing the alignment tensor works for proteins by changing the ordering medium can change the alignment tensor of proteins. It's not easy to do, but it's possible. But because proteins have non-uniform charge distribution, you have little positive patches, negative patches. So if you have a charged medium versus a neutral medium, you can kind of alter the alignment that way. There's been lots of work by people like Christian Griesinger showing that. Nucleic acids are problematic because they have a uniform negative charge distribution. And so you're not going to be, and it very much matches the shape of the molecule. And so you don't have another dimension to do the alignment. Okay, so it's a great idea, Joshua, but it doesn't work for nucleic acids. What could be another way of changing the alignment? If you lengthen the helix, I don't have this here. I will come to that. But later, so if you can't change the ordering medium, the next best thing is to change the solute. And here it's easier to change in RNA the solute than it is potentially for proteins. Although there's some, I'll come back to that. But what was the other mechanism of alignment that we discussed? What's the other way of aligning molecules besides ordering medium? Magnetic field alignment, right? So the magnetic field offers another approach to align molecules, and we'll come back to that in a second. Now, here's a real example of real data. I have 2 RNAs. I've got the RBCs measured on the RNA pieces according to secondary structure. And what you can see is the RBCs measured in the lower helix are attenuated, generally speaking, relative to the RBC measured in the upper helix. And then in particular, those 2 residues have very near 0 RBCs. So what are 2 reasons for why RBCs are 0 in the bulk? So remember we said that if you have at the limit of isotropic motion, you're essentially not aligned. Those bulk residues are effectively not aligned. So the RBCs, if you have an isotropic internal motion, will go to 0. So generally, the dynamic regions tend to have average RBCs closer to 0. What would be the other reason for them being 0? The bond vectors are near the magic angle. The problem here is that I've got 2 different bond vectors, and they're both near 0. And it's impossible to put two non-parallel bond vectors both simultaneously parallel to the magic angle, although they both be kind of close. And so normally you have lots of apartments or residues in a region that are all near zero. That's probably the better model there is that, and since they also have flexible linker, but of course you have to do independent measurements to verify that. The other interesting observation is why do you think the heat sinks here, all of the this is like all the RDCs here are smaller than the top? Why would that be the case? On average, let's say a lot of these guys to be spanning a smaller magnitude than the upper one. Why would that be? And a lot of these vectors have different orientations, right? In the helix, they're all in different directions. Why would the magnitude of RDC be smaller than here again? Who's more online, the upper helix or the lower helix? Based on the RDC magnitudes. So you would say, oh, the upper helix is more aligned than the lower helix, and what could potentially cause that? The length of the stem. So let's imagine for a second this is a longer stem. But it's not true, but effectively it is. It's apparent volume is bigger, let's just say. So then what has to be true about the relative disposition of the two helices? Are they rigid relative to each other? There's interhelical motions, right? So the upper helix is swinging around collectively as a unit around a flexible linker. Now, why is it a longer helix? That's another lecture. But effectively, this helix volume sweeps more of the lower helix than the lower helix sweeps the upper volume. So that it effectively is the longer, bigger volume. So the alignment tensors, as you said, don't match. So the degree of alignment of the blue helix is higher than the red helix. And the asymmetry parameter, which is another parameter describing the isotropy alignment, is very similar. So based on these ratios of the degrees of alignment, we can get a sense of the amplitude of the interior motions by simply taking the ratios. You can get something like order parameters describing interior dynamics. Now, remember, of course, what I've been describing so far is we are solving for the alignment tensor by taking the RDCs and the known structure of the A4 helix. However, as we discussed in class, if I know the structure of the helix, or if I know the structure of my solid helix, and let's say it's well behaved, I can in theory predict the alignment tensor from a simple theoretic model. And that provides a very powerful approach to predict what the RDC should be given a model. Whenever you do analysis, it's always great to have a model, to be able to test a model by predicting the NMR observables, because then you can compare them to experiment and either accept or reject the model. So being able to predict NMR parameters rigorously from first principles is a very important thing. Actually, it's pretty complicated to, so what parameters are easy? We're talking, what NMR measurables are easy to predict, quantitative? Or maybe better ask, what is a very hard thing to calculate that we measure all the time with NMR? Although we're going to go over that because it is quantum mechanical calculations and a treatment of the conformational ensembles and so forth. And it's quantum, so difficult. Great example. What else? What are order parameters? Can I predict order parameters? If I have a model, like an MD simulation model, can I take the simulation model and predict the order parameter? Oh yeah, okay, so order parameters are basically the circle harmonics that I showed you. So the peak loss split minus number 2, when you start with an effective average orientation for the vector, where the angle now is the theta between the average, let's say, orientation of the one vector, and all possible, orientations. So, okay, but anyways, that can be calculated quite easily given a model or reasonably well. But then if you want to calculate R1 or R2, and then at least that needs time scale information. Like what is the rotational overall tumbling of the molecule? What is the time scales of the internal motions? That can be, so it's on one hand very rich in information. On the other hand, you need to know a lot about the system to be able to predict the data. And so it's always interesting to think about the degree of difficulty, like the CSA, RCSAs are very hard to predict. You need to know the CSA tensor and how it varies from site to site, and then also how the molecule is moving, and all of those parameters to predict the actual observed SCSAs. The nice thing about this is I don't need to know anything other than the overall shape of the molecule to predict, in theory. If I have a static molecule, I don't care about its correlation time. I don't care about some spin physics or quantum phenomena. Just need the shape and I calculate the RDC. So that can be powerful because then I can, given the model, predict the RDC and compare it to experiment. So here's an example of this RNA, the same RNA that I described. On average, the experimental alignment looks like this, and the predicted alignment comes from a scale calculation, which is applying a steric model for alignment, looks pretty good. And generally speaking, for RNA, the steric model works quite well to predict the overall alignment of the molecule based purely on a steric model. Now, RDCs help define the global structure. If you're solving a structure of an RNA, having RDCs really helps constrain and define the global structure. You can see that without RDCs, local NOE distance constraints don't do a great job of determining the disposition, the orientational disposition of parameters. But if you include RDCs, you can really improve the global definition because RDCs are long-range parameters that tell you about the orientation of fragments regardless of the distance of separation. Now, I want to come back to magnetic field induced alignment. Remember, we mentioned that you can also use RDCs by just applying very high magnetic fields and then measure the quadratic dependence of the splitting and extrapolate to zero field, you get scalar coupling, and then from that to the splitting observed at high field, you can get a residual downward contribution due to magnetic field alignment. And to do that, you have to measure multiple field dependent values. You have to measure the splitting at varying fields, something that we can do readily at LICC. And then from that, determine the magnetic the RDCs. Now, the nice thing about magnetic alignment is that, remember I mentioned that it originates from the summation of small high tensors for each phase. So each phase has a magnetic susceptibility, the value of which we actually know. And therefore, if you give me a structure of an RNA where you define the orientations of all the bases, I can take those bases and the high tensors and sum them up to compute the overall magnetic susceptibility tensor, which in the case of magnetic alignment represents the alignment tensor. So for magnetic aligned systems, the macroscopic magnetic susceptibility, that magnetic susceptibility is the alignment tensor. And so that's also very useful because you have a model for how to predict the alignment of an RNA, the alignment tensor, and therefore I can predict theoretically the RDCs I should be measuring given a structural model at a given magnetic field from those values. Now, does the magnetic susceptibility, we assume them to be constant, like the guanine, cytosine, thymine, uracil, adenine? Now, do those change depending on structural context, and do we assume them to be fixed? Maybe, but it's an interesting other application of magnetic field alignment is that you can really create models that can predict the RDCs, and then maybe learn something about magnetic susceptibility and kind of find them. Of course, if you are a residue that is flexible, you have to sum up it's a tensor summation, so that residue will have to be, its contribution will be average. It will have an average chi tensor, so to speak. So a residue that's super floppy contributes very little to alignment. So that in a way, the alignment is attenuated by the virtue of the motion because the physical interaction is minimized. So it's an interesting. So you can express the dipole coupling in terms of chi tensor, who itself depends on the base orientations. And these are the values. Yeah.
Don't make much use of 1.2 gigahertz machine.
I'm about to show. Well, that's why I'm presenting. I'm hoping somebody in this room will give back. I think in Germany, Michael Sattler did a 1.2 gigahertz measurement of an RNA molecule and got something like 8 hertz RBCs for the speech things. Yeah. So these are the wonderful magnetic susceptibility tensors in units. The units of susceptibility are meter cubes per volume. And these are the numbers that are measured by the continuity effects. So there's a whole area of measuring magnetic susceptibility. Okay, so here's the thing I want to show is that your magnetic alignment, and this thing was done many years ago before the 1.2 gigahertz. It's interesting that it's 5 at 1.2. You can see that this is the degree of alignment magnetic field alignment is a function of both the magnetic field strength and magnetic alignment grows as a function of what? B, is it linearly dependent or B squared? And RCSA grows B cubed. And of course, the more base pairs I have, the larger the macroscopic chi types are because I keep adding more little i's to sum up, right? So this is the idealized duplex with some assumed internal motional order parameter. And you can see that this is the optimum degree of alignment. The 10 to the minus 3 that I mentioned is, what we really would love to have a sweet spot. And what you can see is that already, for 14 base pairs, we can get a high enough field with 1 to 2 megahertz, we can get close to the optimum degree of alignment. So increasing, this is it. So therefore, increasing the magnetic frequency at 1.2 megahertz, we can almost achieve optimum alignments for nucleic acid size of like 60 base pairs, and then without using ordering. Okay, so now, keeping with RBCs and applications to nucleic acid, Joshua's going to show you how you assign. All of this is contingent on assigning your RNA molecule, obviously a DNA molecule, and Joshua will show you how to do that in his time, his limited time, or his video will be more advanced stuff. But I wanted to show you something more modern applications of RBCs, because this is now, I think, an important area of interest not only for RNA but proteins, and that is the marriage of NMR parameters of all sorts with computational models to generate atomic level descriptions of ensembles of molecules. So our proteins and RNA and DNA molecules don't form one static structure, but they form an ensemble of different conformational states, integrating at time scale spanning 12 orders of magnitude in time. And we would love to be able to gain insights into what are the members of the conformational ensemble, what do they look like, what are their properties. And we're still nowhere near being able to atomically resolve, every member of 10,000 member ensemble. But we are at a point, thanks to NMR and the power of NMR, to at least gain some insights into who those members are and what their characteristics might be. You can think of this as like low resolution ensemble, where we're taking small steps towards that. And the idea is, of course, NMR alone doesn't carry information, all the information required to build an ensemble. Even though it's so rich in information, there's no way for purely NMR data only to really generate all the variables. So we have to use computational models to fill the gap. Computation is wonderful because it can provide lots and lots of information. And for example, we can run an NMR simulation or whatever computational model you like, generate a library of all the different conformational states a given molecule can assume. So the idea is I take my molecule, I use a computational model to enumerate the landscape of possibilities of that molecule. And that landscape contains things that might be, artifactual. I'm drawing it in gray. By artifactual, I mean like not highly populated in the ensemble, like maybe the populations are 10 to the minus 12. So populations that are kind of non-physiological because maybe they're artifactual. But others are actually dominant states in the ensemble, meaning they have more appreciable populations, and therefore are things that a cell might encounter, like in the concept. You might actually see that combination. So the idea is generate the library knowing that it's got bad apples and good apples, the good apple color, and use experimental data, of which NMR really is the most powerful in terms of having atomic level information about the averages of the ensembles and extracting out your ensembles this way. And then of course, when you get the ensemble, you're obviously overfitting. This is a natural part of the beast in this type of analysis. You have more unknowns than you have measurements. So whatever you do, this model is by definition going to be underdetermined. And so you want to do independent tests of the validity of the model. Now, I want to quickly defend, so I want to first point out this underdetermination problem. The goal is not to be of course, you'd love to be overdetermined. But what I would want to do at the end of the day is generate a model that is closer to truth than any other model, given a set of data. right? Like that's the best I can ask for, right? Is given a set of data, generate a model that depicts reality as much as possible, as best as possible, relative to anything else. That's the only thing you can hope for. And so whenever you see this and somebody starts yelling about overfitting, you have to remember that the current depiction of the molecule as a static structure is itself also a major sin, because that's definitely wrong, right? And so you could have a debate about how bad is this model, given that it might not be perfectly sampled in the space, but at least it gives some credence to the dynamics and maybe captures aspects of it relative to a model that is completely ignoring these motions and simply showing a static depiction. And in fact, the static model, as you'll see, will often fail to explain the NMR data because the NMR data is ensemble average and requires an ensemble description. If my RDCs are sampling a bunch of orientations and you get a dynamic averaging of the RDCs, if you were to now insist to analyze those RDCs in a single static structure, you're going to get the wrong answer. Let's take a simple example. If I have a bond vector in a loop that's super dynamic, what will the RDC be? 0. If I take that and I shove it into a conventional structure calculation program, which requires that there's a single static structure, where will it put that bond vector? You're only allowed one structure to explain the data. So which orientation will it give for the bond vector? The 0 RDC bond vector. The magic angle. That's the only way, right, to explain. So you'll end up with an artifactual model that says this highly dynamic residue has its bond vector oriented along the magic angle, versus the model that says that in fact it's super dynamic. Now, of course, the challenge is how do you distinguish these two? But it's very important to appreciate that this always happens. Let's take NOEs. NOEs go as the, you know what NOEs are, let's say. When I assume you know what NOEs are. But they depend on the internuclear distance to the inverse R6. Suppose 50% of the time I'm 3 angstroms apart, and 50% of the time I'm 6 angstroms apart. And Joshua, you better feel the answer to this. What is, and then I measure it, but the NOEs, I get one NOE measurement, and I try to, again, interpret the NOE as a static model. What distance will I end up computing? But average over what? Average over R to the 6. So it's going to be biased to which distance? Shorter distance. So if I just did like my NOE depends on 1 over R6, I would deduce a distance of 3.5 angstrom. So not only is it not 3.5 angstrom, it's never 3.5 angstrom, right? It's either 3 or 6. And yet your structure is going to be artifactual because it's imposing a distance that actually means 7%. So whenever you think about these approaches, you should appreciate the danger of overfitting, but you should also appreciate the danger of underfitting, because you're now imposing a static model on something that may not be actually perfect. So there's a wide range of data that can go into this, but what's required is I need to be able to compute the data from this for every conformer. I need to be able to predict the NMR observable, so that I can tell which one should I include in my ensemble. And I just told you RDCs are because you can predict them. And the reasons you can predict, although those also depend on distance, on dynamics, time space. And the reason is a little complicated because to really predict an NOE rigorously, you need to model the spin diffusion, you need to know the time scale, the correlation times of different protons, but it can be done. And using approximations. And of course, scalar couplings can also, there are empirical parameterizations for that, and so on. Okay. So I just want to show you, there are many different models that can be used to generate the library. But as we discussed, and as Ethan's idea, that it would be great to have many RDCs in this exercise. Since we are having the problem of the ensemble needs more variables than we have measurements, it would be great to change the alignment. And as I said, treating the ordering median isn't practical for nucleic acids, but you can change the nucleic acid itself. You can change the, for example, here's an RNA. You can elongate the lower than this. So now the magnetic field will be on average along this direction. here it's changed a little bit. If I elongate the upper grid, then it goes that direction, and then and so I can be variable elongate. So in some, and by the way, in proteins too, you can anchor them, giving your protein bind to some other thing, and then have its shape change. The key is to change the shape in a predictable way. And in the case of RNA, we can subject all of these elongated constructs to Pale's calculations. I really care just about this thing, but and I measure RBCs across all of these. When I generate the ensemble of this, I can predict the RBCs here by simply appending a helix and elongating before I do the Pale's calculation. Right? So again, I can measure the predicted RBCs. And as we discussed, these are the directly bonded CHs are the ones you typically measure, as well as NHs or whatnot. So NH and CH RBCs are the ones that give us the maximum signal certainty. And so let me show you an example of this. So let's say I run an ND simulation on a force field. This is an 8.3 microsecond. This is the relatively long simulation of this RNA. For every snapshot in the simulation, I can subject it to the Pale's calculations to predict the RBCs. And then I average over the entire conformers in the simulation. And so this is the predicted RBCs. The different colors here represent the different motifs. And then the measured values, and you can see the agreement. So here, one of the great things about RBCs again is that given a model, here it's an ND model, I can predict the RBCs, average over the conformation, then compare them to the experiment, and the error in the measurement here is about 2 hertz. And so you can see that the RBC deviation is, although there is a correlation, the magnitude is way greater than the uncertainty. Now, the trick that we use to generate the ensembles is to pick subsets. So imagine you have good apples and bad apples. What we're going to do now is take the full ND generated conformation and ask the question, is there any one structure that satisfies our RBCs? But if there was, in fact, a single static structure in the ND pool that satisfies my data, there's no reason to go to a more complicated model. You would basically say, this is the model that best satisfies all of my data. It's just one of the structures, and maybe this is a static R. But if I look at the solutions that I get for the one ensemble size of one, the best case scenario is still way above my error. So out of tens of thousands of conformations, none of them, the best of them, gives me still a very poor agreement with the RBCs. So the next best thing is I say, okay, imagine I have a 2 conformational states that dominate the data. So I say, let me try every pairwise, like every 2. And there's a Monte Carlo sampling scheme to make it sufficient. You don't have to go over that. But the idea is, can I find a 2-state ensemble that satisfies the data? And not surprisingly, you do better, but it's still not enough. And then I ask for 3 and 4, and I keep increasing the ensemble size until I get close to the error. And usually it plateaus. And the idea here is you want to, as soon as you reach experimental error, you want to stop increasing the size of the ensemble. And then let's even pick 20 here arbitrarily. So there's 20 conformations that are sufficient to average the RBCs and make them come into agreement with your data. Now, it doesn't mean that there's only 20 conformers in the ensemble. How many conformers are in a typical ensemble of proteins there? Like, you give this.
For NMR data or for real life?
Just analyze a protein in real life. How many conformational states do you think it samples over like a second? You know, is it like 10, 20, 1,000, million? 100,000. It's like a million. No, it's astronomical, right? It's like tens of thousands. You know, think about every side chain, rotating, every unique conformer. There's thousands of these, right? So you're not. So when you look at 20, that 20 number is, this is the number of states required to accomplish the averaging required to satisfy the data. And the way I like to think of it is, imagine you have an S letter, and ideally I would draw the S completely and have the nice line defining the S. But this is an approximation to the S, hopefully. And what you have is a dot here, 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 a dot here. So you have little dots that capture the essence of the S, but that's all that's required for the data. The data is not in, you can't specify the S uniquely. But if you repeat this analysis many times, you get slightly different ensembles every time. they differ. They're not always, if you were to do this analysis many times, each time you capture a slightly different ensemble. But if you look at the ensemble, it always traces an S in some of its features. So we don't know what it really is completely, but we're getting a sense that it looks S, it has S-like features, for example, or an M or an N. And in particular, in this case, features related to orientation, which RDCs can define. So the name of the game is to appreciate that your ensemble is incomplete, it's underdetermined, and you need to be very smart about not over-interpreting it, and seeing some of the features. Of course, every time you reselect the ensemble, you get a completely different level. Like, in one case it does S, and then it does M, and then you are obviously, there's a large swath of solutions. But normally you'll get an S, but with slight deviations, if you have it updated. Now, so if you do this, so this is what it looks like. So now, if I do a ensemble 20, I do better agreement, I get better agreement. Now, if I use another model, this is an MD simulation, this is structure prediction model. This is the model of the library generating structure prediction. These are not important here. But the end result is only as good as the input, right? You input crap, you end up with crap. If your MD simulation is not sampling space properly, no matter what you do, you're never going to satisfy. Hopefully, you will have a hard time satisfying this. This is a better structure prediction model that has better sampling, and now we get a better fit to the RDC. So a better starting point. But the structure prediction is much easier to generate for RNA than the MD simulation because it's super fast. This is a Rosetta model that's called a POC project that has been developed by Richard Gas and David Baker. Okay, so now that you have, so if you're a student and you say, oh, look, I have this, I have myself an ensemble, it's fitting the data, you still have to worry that you are overfitting and you need an orthogonal test. And so the orthogonal test often uses chemical shift. So chemical shifts are, again, one of those parameters where the calculations are not easy. Well, not easy, but the chemical shift is a quantum mechanical phenomenon that deals with the electrons and their distributions around a given nucleus and how that impacts the shielding. What does the chemical shift depend on? I'm a nucleus and I have a chemical shift. What dictates my chemical shift?
Magnetic field.
What?
The magnetic field is field.
So the magnetic field that I feel, and that's shielded by the electrons around me. But what determines the shielding of how much I get shielded?
Everything.
Like the geometry of the bond, the torsion angles, the presence or absence of solvent, hydrogen bonding, am I tautomeric, am I protonated? Lots of things impact the chemical shift. And even then, if I want to predict the chemical shift, one must carry out a quantum calculation. So typically, you will have a quantum fragment. So you'll take your nucleic acid, and the fragment will be subject to high-level quantum calculations. That's the QM part. And then the rest of it will be involved in molecular mechanics. This is to make the calculation efficient. Otherwise, performing quantum calculations of the entire thing will take forever. Fortunately, a program called NMR, by David Case's lab, allows you to very quickly take a given RNA and produce those fragments, which you can then just simply submit to the DFT program. So it really helps do all the front end. And of course, now, if I have an ensemble, chemical shifts like RBC, there are also ensemble average over many conformations, over the ensemble of conformations. So one will have to do this calculation for every member of the ensemble, and then average the chemical shifts, just like we average the IDCs, because it's an average measurement. You don't want to end up with a 3.5 angstrom problem. You want to calculate an average over all the ensembles. So I want to show you, I want to convince you, this is the best proof of dynamics in nucleic acids. So if you look at DNA, this is DNA, and I'm not going to tell you, an ensemble model is generated for a DNA duplex, and that ensemble has a number like that, maybe 12 members. If you take the chemical shifts of the sugars and you predict them using a static structure. This is a very high resolution NMR structure with an NMR that was tabular, that was obtained an enormous amount of information. And yet, when you predict the chemical shifts, you can see that for C1 prime and C3 prime, you do a horrible job from that static structure. This is now the sin when you interpret the data using a static structure, like the 3.5 angstrom sin. Despite the fact that this DNA was determined with the largest number of conceivable NMR parameters, the static structure, the DNA fails to do the simplest thing, which is to predict its chemical shifts. So then you look at data like this, and most of the time, for many years, people thought, oh, DFT sucks. Those chemical shift predictions are.
The program calculated the CSA insert power to extract the chemical shift.
It effectively is titrating the CSM. And then the isotropic part is what you spit out. So, but yeah, those are DFT calculations, which I'm sure you guys will do. In Anne's lab in particular, to get the CSA tensors. It's the same approach. And so normally you get this and you say the calculations are not working, throw away these quantum calculations have some issues. But of course, especially with sugar pockets in DNA, this has been known for centuries, for decades, that the sugar pockets are interchanging between C3 prime endo and C2 prime endo. Predominantly C2 prime endo, say 70%, and C3 prime endo, say 30%. And we also know that the chemical shift of the sugars depends on the sugar pocket. So if you've got a 70-30 mixture, you will never be able to satisfy the chemical shift by assuming one static state. It's back to the 3.5 angstrom problem. So if we now come in and say, let's use an RBC ensemble instead of a static structure, so now you're seeing the prediction coming from an ensemble generated using the approach I mentioned, where RDCs were used to build an ensemble from the MD simulation. You can see much better reading. So in our world, I would argue that this model is a better model than that one. So this static model fails to predict the chemical shifts. This model predicts the chemical shifts better. So therefore, I will embrace the ensemble model because it explains more of the data that I have. Does it mean the ensemble is perfect? No. Does it get the sugar pucker distributions correct? Very close. In fact, so now I can look at the ensemble and say, oh, what are the sugar puckers? Oh, the sugar puckers seem to be 80-20. And that's the S you draw. Now, is it 80-20? No, there's probably many other sugar puckers in addition to the CT parameter, the CT parameter. But that's getting to closer details, right? But at the level of saying sugar puckers are not static, they exist in a distribution of two conformations minimally, that's an advance. And now good luck every time by any method other than NMR. I mean, it would have been impossible, I would argue, to definitively demonstrate that in DNA, your sugar puckers are constantly looking back and forth and get those distributions. So in RNA, we do the same thing. So in RNA, it's even more insulting. Well, the MD simulations and do very poorly with all of these. And you can see that the NMR derived such a
Hey, Ashim.
Oh, sorry, I didn't notice you're there.
Okay, no, enjoying the lecture. Listen, I was wondering for the predictions you had on the previous slide, and I'm assuming for the sugar, part of what's driving this is a global property of the ring so that these would be correlated, like the changes in C1 prime and C2 prime, would probably be somewhat correlated. And in a 2D experiment, you would see a non-Gaussian interesting peak shape. Does that make sense?
Yeah, that's interesting. So you're saying that the chemical shifts of a given sugar, C1 prime and C3 prime, have a certain distribution between them because they co-vary together.
Yeah, like a pseudo rotation or some dominant global feature of the ring. Does that make sense? Is that likely?
Absolutely, it's called the sugar freeze angle. So there is the sugar pucker can be defined by a single sugar freeze angle, and it defines once because it's a closed ring. So once you have that one parameter, it defines the torsion angle around the sugar. And that creates correlations because of that between the different total levels. And as a result, you end up with, you would predict, I think, based on what you're saying, correlated change in chemical shifts, which we actually did not look at, but that would be expected.
Yeah, we could think about either, freezing it to identify individual members of the conformational ensemble. And this would make strong predictions about what you should see. And then sometimes a very controlled lyophilization can give you a similar result, but the freezing is just so much easier. It's just a thought.
Now, that would be great. And in meeting with Anne, she asked the other question is, I didn't mention this, but in these simulations of RNA, it's very hard to change sugar buffers. Once you have an initial sugar buffer, MD doesn't know how to change sugar buffers. It's just stuck there. And that's why those other models are better. But the question that arises is, like, what dictates the barrier to sugar recovery? I actually don't know. Anne asked that question. And so what exactly are the time scales? We think they're nanoseconds. They don't give rise to chemical exchange. So they're faster than microseconds. But what is the barrier? What is the cause of the barrier? How does it change depending on sequence and all that is, I think, I hope about it. And yeah, and it would be great to do this all experiments to freeze out the members of the ensemble and figure out their identities, because I'm sure it's not just C2 prime endo and C2 prime endo, you also have C4 prime exo, and like in A tracks, for example, in certain motifs, certain sugars are more prominent than others. So I think that would be great to explore it.
Yeah, and we've set up all the different experiments. So as soon as you have a molecule, we were also, for an analogous reason, on the peptide conformations, looking at zero quantum and double quantum, correlations and all that stuff. But we could just start out with a chemical shift, simple chemical shift correlation between neighboring carbons. Yeah.
Jeff will just painstakingly assign his sugars, and he will be presenting how he assigned his sugars, and then we can take that sample and build that collaboration. I think it's great. And by the way, what I'm not showing you here, and I'm sorry not to have it, is the spectacular failure to predict phosphorus transition. So really, we were hoping that the ensemble model would actually fix the phosphorus predictions. But the phosphorus prediction, I don't know why they thought that. We have no idea what's going on with the phosphorus. The ensemble does not rescue phosphorus predictions, and maybe there's some salt effects, the modeling of the salt, and so that's, I think, an area that is very fascinating to pursue in the future. Working with David Case to still actually work with on some of these calculations to see if adding metal ions in the vicinity of the fragments might shape the phosphorus transition, but complete failure.
It's interesting. Interesting.
Yeah.
Literature comparing crystal structures and solid state phosphorus data really strongly emphasized the effects of hydrogen bonding on the various PO bond lengths and tried to make kind of like a principal component analysis out of that. But I think we could do a lot better these days.
That's another area that we could talk about.
Yep.
I'm sorry, they're not the process. It looks really horrible. Okay, so this can be done pretty efficiently. I'm just showing you some examples here. So the nice thing of this approach is that there's lots of NMR data in the public domain, in the VMRV. And so one can take RDCs that are already published. There's about 64 RDCs data sets for RNA structures, and their chemical shifts are also available. And next time we have this class, and I had this idea too late, would be to have you guys, for an exercise, run the pipeline to render some of those NMR entries into useful ensembles, and then doing the DFT calculations to get the chemical shifts and tests, and then we could publish the results from the class. There's 64, so that we have like 4. If we put away, a subset every year, we can help build up the entries of the ensemble entries. And again, I'm not saying these ensembles are perfect, but I am arguing that one can test whether they are a better representation of the data. They better account for the data, and perhaps better represent the reality of the molecule relative to some of these set structures. Okay, so Joshua, you're getting a little more time if you want to get started. So I was going to just maybe go quickly over how we make nucleic acid samples. I don't expect everyone here to care about this too much, so I'll go through it quickly, and then Josh will come and show you how we assign those samples. So for RNA, so basically for both DNA and RNA, there's two ways to make your NMR sample. There is enzymatic approaches where you let the enzymes do the chemistry and put your molecule together. And the other is using chemical synthesis. And each of them has their pros and cons. And so for enzymatic synthesis of RNAs, we usually use a T7 promoter or T7 RNA polymerase. We have a piece of DNA that encodes the RNA sequence of interest. You basically want a transcription reaction where you feed the T7 polymerase the DNA templates, and then the NTPs, magnesium, and then everything that you need to have, and basically make your RNA. I'll share these slides, but there's pros and cons. Smaller, shorter RNAs are harder to make this way. There's sometimes bending ends. The yield can vary depending on the sequence context. So here's the polymerase during the reaction. There are ways to get it purified. So when we make RNAs, often you end up with bending ends like I+1, I+2 products, because the polymerase continues to add at the ends. And if you want to really purify your RNA, you can add a ribozyme, which cleans itself out to get super pure products. We don't need to worry about that here because NMR doesn't get affected by the bending end, but it's purifying for crystallography. You don't have to worry about that. You can do there are corresponding approaches to making, and by the way, you can label your nucleotides using uniformly labeled carbons and nitrogens. There are some NTPs that are available that are site-specifically labeled for both RNA and DNA. There's also enzymatic approaches for DNA synthesis involving PCR or primary extension. Again, same idea with a polymerase. You generate more molecules by meeting the polymerase. You can uniformly label the whole sample. And there's a variety of approaches for primary extension of DNA to create the labeled material. And then, of course, you have to purify it. Then there's the chemical synthesis approaches in phosphoramidite chemistry, or 3' first you make iron, enzymatically you make nucleic acid 5' 3', chemically you make that 3' 5', and here we display phosphoramidite chemistry to basically take a machine, you tell it the sequence, and it will template out your nucleic acid, RNA or DNA. We have a synthesizer, by the way, so if you ever wanted to use it, you're welcome. And the advantage here is that we can incorporate nucleotides at specific positions that are labeled. So we could make it like for solid state applications, for example. We could make this thing about 60 nucleotides easily, right, John? 60 nucleotides?
It can be up to like 90 or 100, but even better.
Yeah, so let's say 60 very good. So like, so then one can label individual nucleotides with specific carbons labeled or specific carbonyls. That's really powerful in terms of assignments or spectral simplification. You can also incorporate all the modified nucleotides like M6A, 2' methylation, there's a wide range of modifications that are sort of in nature, and you can synthesize them. Many of these are available, but they're also labeled. You can buy custom synthesized isotopically labeled like M6A and 2' methyl. And typically purification is done using polyacrylamide gel electrophoresis, which is a massive gel to purify your nucleic acid. And then you cut it out of the gel, and there's various ways to extract the RNA or DNA from the gel, whether you use crush and soak or electroelution, where you basically like elute out the nucleic acid from the pieces of the gel that you have. And then you can also run additional HPLC purification to further clean up the RNA sample. Here's the synthesizer. Back in the day, the Duke, this has been moved to Columbia now. But the synthesizer can be used. Also, we can do multiple syntheses at once. So at this point, we can do 16 samples. There's 16 channels we have. 8? What? 12? So we can perform 12 syntheses now. Right? Yeah. So we can make 12 different NMR samples with different label size and multiplexing.
And we have tried for
wells for the outlet, so up to like 20 more feet.
Right? So, and typically when we make our samples, there are things to worry about, like you often will have some acrylamide contamination. We have an inventory of all the things to watch out for that I created to your sample due to imperfect purification. From polyacrylamide to ETA to various metals that can help you figure out, and then you might have one of desalt alcohol in some cases, but more or less, these with the key take home is you can make nucleic acids very easily, and sometimes you can even just buy it, including related size. So there should be no fear at all, no excuses not to do NMR nucleic acids to the Amazon, especially the gray hair. Samples, and there's super quality for every care. We can produce them at high yields, and we can produce lots of them. So it'd be great to work together on that. So now, Joshua, would you like to come up and walk us through your assignments of distributors? So for everyone in Zoom, give us 2 minutes to transition because Joshua's going to connect us to you.
Since you can make DNA at such high yield, which slipping can you do at natural organisms?
Oh, so actually, thank you for the question. Actually, I forgot to mention that in solution NMR, you can measure RDCs. We measure RDCs in natural abundance. We don't bother labeling.
And if it's
As I mentioned, yeah, some issues. So we do carbon, we do CHs. 2 millimolar samples of the 900, plenty of sensitivity to do RDC measure. HSQC, no worries.
right.
RO test, that's much more difficult, then you need the latest. But you can do all the RDC measurements, which is why we have this idea of generating ensembles for every possible anywhere. DNA duplex to get it to sequence dependence, because that's actually with the facility with MISUC possible. I can probably kill me for not probably all the NMR machines and get something like nucleic acids, but you can do it in natural abundance, super nice. In fact, the ensemble that I showed you was done in natural abundance. Same for RNA. You can measure the RNA in natural abundance. In fact, it's nice because you get rid of carbon-carbon couplings.
Yeah.
So some of the HSQCs are actually, you don't need to worry about constant time.
Yeah.
So you actually get back some sensitivity because you don't need to use constant time experiments. Yeah, it works great.
While Gotcha was getting set up, they wanted to point out the completeness of many of the mathematical problems of two domains with alignment are similar to the problem of rotational diffusion of two domains that are linked together. And although the same mathematics come to bear, because again, you have two tensors, and you're wondering whether they can be superposed. Now, one of the good things about RECs, as we told you, is you can measure a lot of them. The rotational crumbling, you're measuring relaxation rate.
Yeah.
And it's a lot harder to get that number of and that kind of precision. It's available from
Yeah.
RECs. But it's not built which one was for example that from very historic. And Michelle Gill, when she was in college, the freshman did, and I explored it, trying to figure out how we would ever analyze relaxation for time dependent changes in learning patients.
But you understand and you have a paper that it's one of the treating the
Oh, yeah, a long on that.
So first thing.
But we haven't had a follow-up paper.
Yeah, that was because that's similar to the kind of the everything alignment where you can there's a piece of copy. But that was some relaxation. Yeah.
Yes. Yeah, so. You have one? You have one too? Oh, how it goes.
Oh, you know, other parameter that the maximum unification. So
Yeah, it is generalized. You already know.
It's very similar.
Exactly, because they both reflect the.
Yes, they are very similar.
Spherical commodity.
Yeah, you can be heard of like.
Generally, if you give one of the describe
You'll be fine.
actually is mathematically constructed the following way. Imagine I have a sphere and I have one vectors in every possible orientation. What is the average RBC? What is the average of sphere harmonic? Square, so that you get rid of the minus sign. So to basically to quantify the degree of alignment of a molecule, the answer is, well, if you ask the vector, then they're all each aligned slightly differently because they're oriented differently. So to get an unambiguous mathematical structure, you say, I'm going to average over all one of them in that parallel curve. And that's the generalized degree of order definition. But as I said, that they're all sort of at the end of the day, it's all the same. E to 0 or y to 2, sort of the marks. But you know, I met R at an ENC poster, where you were calculating everything from order time. And you can develop a model for the volume swept or the area swept of the cone. And I think that the fact that it was coded as zero orientations of the constructed generalized degree of order.
Okay. Good to go.
So I'll just present a little bit on RNA and DNA assignments. I'm not going to go through like full complicated assignment, but I just want to talk about like some of the patterns that we use just so you can get started on it. And then I'm just going to talk about these in the context of unlabeled samples, because you label them, you can pretty much get your assignments immediately, but unlabeling is probably the most common strategy. And then probably just gonna limit the discussion just to DNA for some reasons that I'll talk about later. But okay, to remind everyone, these are the monomeric units of DNA, and RNA. This doesn't have thymine, of course, it has zero. So I'm just gonna be referring to like these nucleotide numbers a lot during my presentation. I'm sure, well, everyone in the machine lab should know these, but for the protein people, this is just like my reminder to you. Like basically, anytime I refer to something as a prime, it's going to be on the sugar ring, and it looks the same in every nucleotide. But anything that doesn't have a prime is going to be on the nucleobase portion of the nucleotide. And by convention, the IUPAC convention is the B hydrogen attached to the amino groups of guanine, adenine, and maybe and cytosine are the ones that are involved in the hydrogen bond between hydrogen bond and an acer. And then I'll talk about some of the sugar chemistry later. But when you just have your DNA, after you've made your DNA RNA sample, like the sheet just explained, you of course would want to run a 1D. And these are approximately where all of the different nucleotides, where all of the different nuclei fall on a 1D spectra. You can see that in RNA, you're kind of unlucky because there's more, there's less chemical shift dispersion amongst the sugars compared to DNA, which is why I'm going to talk about DNA assignments because you can actually do them. But for DNA, since they are more dispersed, and you also get this knuckle group, you can pretty much assign the entire molecule with like effort. So these are just the, again, reminder, these are the nucleobases, and then these are the sugars. Of course, in RNA, you have this 2' OH group. In DNA, you have two hydrogens here, and they are designated prime or double prime based on the stereochemistry. And this is just the IUPAC convention, but the double prime hydrogen replaces the OH group in the ribose sugar. 5' is again based on stereochemistry, but yeah, this is what the sugars look like. So if you have your duplex or hairpin, the first place that we start off for assigning is the immunoresonances. And they call here, immunoresonances are found in thymine, guanine, and uracil. And essentially, well, I'll get to this in a second, but we like to start with the immunoresonances because they have the least amount of signal in that region, so they're easy to identify. And then you can kind of use that as a starting point for the rest of the molecule. But I just want to say everything that I'm talking about is actually coming from, sorry for the streamline, but this Wananga review. So in case you want to learn about assignments more deeply, I definitely recommend reading that. As for workflow, it basically, for unlabeled DNA, it uses all the experiments that we talked about, that are talked about in class. We're just using nosy, cozy, and Hoxie. So we start with the nosy, do assignments of the aromatic regions, and we move to cozy and Hoxie to do assignments of the sugars. And then if you really want to do the phosphorus, then you can move on to phosphorus from there. Okay, so back to the immunoresonances that I talked about. So it turns out that the immunoresonances between base pairs is quite close to one another. So no matter whether or not it's on the 3' or 5' end, you will see a cross sleeve between immunoresonances of base pairs directly above and below. So you can do an entire walk along the duplex, notwithstanding the terminal ends because of chemical exchange with solvent. And you can reasonably assign basically the entire duplex by doing this. So just as an example, the G2 is here. G2 should have a cross sleeve with, what is that, D24. And this is T24, and you walk over and then you just keep doing that. I'm sure that there's an analogous process for chain assignments, but yeah, and so you would keep going as well. More. Is the computer do this for you? Oh no, we do this in.
Yeah, the computer does the thing.
It's
I'm
not.
sure if you give it to Claude or
Yeah.
ChatGPT, would you didn't try to assign?
No, I'm
Has
not.
anyone ever tried to have ChatGPT to assign something?
But I think next week Bruce Johnson is coming.
Network that has the RNA assignment group. And also RNA also has it.
But there's an AI element to assigning, like,
Yeah.
it's just yeah. And you should gave that picture. It looked like ChatGPT told it assign it. Would it do it?
Maybe for very small constructs, but my guess would be, since we work with a lot of overlap, so you kind of have to use your human intuition to really think like this is probably overlap with another.
You can ask AI to get English.
Right. So, once you have your immunoresonances assigned, which is going to be H1 and H3, then you move to your amino groups. You can see that in a GC base pair, there's going to be nosy crossfades between the immuno groups of guanines and the cytosines amino groups. So that's basically stepping across the strand. And then once you cross into the aminos, you can get into the C8 and C6 region, and this is called the non-exchangeable walk. And this is probably the most important, or the most extensive assignment task that you might be required to do for nucleic acids, which is once you get from an amino group to say like a cytosine H6, you can walk to its H1 prime because they're close together in space, and then you can walk from the 5' direction from 5' to 3' to the next nucleotide in the same strand with aromatic region, and then keep going along. So it's basically, since nucleic acids, they're helices, and well-defined helices have very almost conformation independent distances between their base pairs that you can use as a guide to figure out what peaks belong to which base. And then, of course, once you're able to assign a nosy, like what I have on the top, you can transfer these proton assignments into an HSQC. Can be carbon HSQC or nitrogen HSQC, depending on what you need. And then usually, we stop here. But if you wanted to take it one step further to assign sugars, there are now dislogs for example, in DNA, H2 prime and H2 double prime, you can do the same thing. Instead of going from aromatic to H1 prime to the next aromatic, you can go from aromatic to H2 prime to the next aromatic. Of course, this is a little bit more complicated because there's less chemical shift dispersion between the H2 primes, but it is possible. And that is what I had been working on for a while. And then if you wanted to get into further trigger regions in DNA, you're lucky because the triggers kind of resonate in different regions in the spectra. You can use either a nosy or a cozy to go from each of the sugars in the ribose ring. So even though like H1 prime has a crossbeat between H2 prime in a nosy and also in a cozy. But in a nosy, you might see crossbeat between H1 prime and H3 prime, H1 prime and H4 prime, and all of these different regions. So you kind of have to play around using a combination between these two experiments to try to figure out what the correct assignments are. But once you recognize these patterns, pretty much it's time. This is a nice example that I like to go through, which is like the advantages of using cozy and epoxy for assigning the stereo center carbons in the ring. So for C2 prime and for C5 prime, a really neat thing is that you can see like two different sets of peaks for the two separate hydrogens at the positions in the ring. But the scalar coupling between the neighboring protons is different. So the intensity of the peaks and the intensity of the cross peaks in a cozy will be different. It just so happens that H2 prime is in the anti-conformation to H1 prime. So it has J coupling that is about twice in magnitude to the H2 double prime, which is cash. So you can distinguish them from cozy like this, but you can, if you don't want to work with cozy, you can work with epoxy, which basically just collapses these multiplets into singlets, better signal, you get better dispersion, and it's a bit easier to assign. You can also epoxy, of course, you have the long range interactions. So, for example, like region A is cross peaks between H1 prime and the H2 prime. But in epoxy, you have all these other regions as if it was like a nosy, but it's happening through bond instead of through space. So it's more rich in information it's less rich information than a nosy, but it's a little bit more rich in information than a cozy. So it's like this intermediate sweet spot that allows us to assign.
What concentration is example?
So for DNA, since you can just buy them, we just buy a lot and it's cheap. So our samples are usually like DNA samples are definitely above 1 millimolar.
What?
1 millimolar.
1 millimolar.
Yeah.
If 4 millimolar, you start getting aggregate, like they're coming, the correlation time kills you.
Yeah.
So they're gonna tell you that here. But you can never do 2 millimolar.
Yeah. This
Depends on any of the sequence. It sometimes starts to aggregate even at 1.5.
yeah.
But how much does it cost you to buy your sample?
How much does it cost you?
How much is the DNA sample?
It costs to make a NMR sample, let's say like a 1 millimolar NMR sample, it costs you $50.
Sorry, out.
Yeah.
It's a great
No more.
business to be
Yeah. And so
bigger than proton scale.
in the sugars, like H1 prime to H2 prime is about like 10 hertz. The Gauss conformations are about 5 hertz. So in this cozy, like I don't know if you can see very well, but like this peak is definitely the anti-hydrogens, and this is definitely the Gauss hydrogens. There's another stereocenter, which is D5 prime, that has also H5, two protons on it, and they are both Gauss, so they're very difficult to distinguish from.
And you know, if you have a each prime angle, how the coding looks like, do you know that?
So you can measure you can, be quantitative about the velocity and measure the coupling by measuring the split across the couplets. If I remember correctly, the C, when it transitions from C2 endo to C3 endo, the coupling gets bigger.
It switches.
Yeah. And so this coupling is also corresponds with the upfield shifting of the carbon at the C3 prime and moderate C4 prime. So it's also a really good way to distinguish sugar coupling, and which is what we've done extensively for this construct in our lab, the A6 DNA, but we also have another one, A2D. Yeah. And then the last mini set of experiments that I would wanted to mention was the very first experiment that I talked about was doing the nosy walk between the EMI nose, but if you really wanted to confirm hydrogen bonding between at a base pair, because sometimes you might not see the EMI nose because they might be exchanging. They are very weak relative to other crossbeats on the spectra. You can run these J and N protein type experiments, which just detects if there's a scalar coupling between N1 and N3 in a base pair. And it can be very instructive about figuring out, especially if you have odd secondary or tertiary contacts.
You're able to measure those in natural coupling?
With a lot of states. Yeah.
Okay, can you go through these metrics?
Which one?
The next slide.
Oh,
HGDA.
Oh,
Where the AA research come from, the company.
oh, this is yeah, this is an old paper that I just grabbed these structures from. I don't know if these are accurate structures, but
Yeah, both. It's
yeah.
the
I don't think these were not from the paper that was using B-form DNA. I think either HDNA. Yeah. Good process. Yeah, but that's all I have for you today.
Any questions online?
No, for me.
Great.
And if you're worried about whether there will ever be an exam, the plan is to hand out the exam next week after the problem setting.
It's in class or
Take home.
hungry now.
Probably a week or so. Got to go back before the end of the semester. See everybody next Wednesday.
It's good. Are we going to choose papers according to the syllabus?
Excuse me?
I mean, you're talking about exams, so I was wondering from the syllabus, you say we are going to choose papers, but we have to consult the instructor. At what point are we going to do that?
Final exam is just a paper. You write your short paper on any NMR topic you're interested in. So just email Ann or me what topic you're interested in writing about.
Okay.
I'll send an email out to everyone.
Okay, thank you.